Okay, good morning, everyone, and welcome to Grand Rounds. I'm Kader Çetin-Gedik. I'm a pediatric rheumatologist, recently started at Children's Hospital of Pittsburgh after working at NIH for 2.5 years. Today, we are honored to have Dr. Raphaela Goldbach-Mansky speak with us on autoinflammatory diseases and lessons learned from using a genetic approach in this patient population. As a reminder, questions can be posted in the Q&A throughout the lecture, and you can join us for a more informal meet and greet at the end of the presentation by asking the host to unmute you. Dr. Raphaela Goldbach-Mansky is the chief of the Translational Autoinflammatory Diseases section at the National Institute of Allergy and Infectious Diseases at the NIH. Dr. Goldbach-Mansky's translational research program focuses on clinical and translational studies in children with early onset autoinflammatory diseases. Her research applies an integrative approach to characterize the genetic and molecular causes of autoinflammatory diseases and to design targeted treatment studies to investigate the role of specific autoinflammatory pathways in the pathogenesis of autoinflammatory diseases. Her studies in patients with IL-1-mediated autoinflammatory diseases, NOMID and DIRA, established targeted treatments with IL-1 inhibitors as a standard of care, and her recent translational and interventional studies in patients with CANDLE, SAVI, and other autoinflammatory diseases with chronic interferon signatures focuses on understanding the pathogenic role for type 1 interferons in the disease pathogenesis and organ damage. Please join me in welcoming Dr. Goldbach-Mansky today. Thank you.
Thank you so very much, Dr. Gedik, for actually this very kind introduction and for seeing you this way. So it is my pleasure to be here and talk to you about my passion, and that is treatment and discovery of pathways of autoinflammatory diseases. These are my disclosures. I'll first discuss de novo mutations as cause of autoinflammatory diseases and then present 3 cases with a focus on a novel group of autoinflammatory diseases. So autoinflammatory diseases are systemic conditions that are rare, genetic, and multi-system. The immune dysregulation is primarily affecting innate immune system. And tissue infiltration with neutrophils is a landmark of those conditions. While genetic mutations that activate the IL-1 activating inflammasome or pathways that activate the inflammasome cause IL-1 mediated diseases, more recently, conditions with chronic type 1 interferon signatures have provided insights into other pathways. NF-kappa B signaling and TNF signaling dysregulation are another group that I won't talk about, and modifiers of immune responses, amplifiers, are another group of autoinflammatory diseases that I touch on today. Overall, these conditions are rare, with definition of rare less than 200,000. Most of those conditions have less than 3,000 patients in the United States. Most are actually in the realm of less than 500. This actually points to a problem of rare diseases. Rare diseases in general are common. There are 30 million affected. 40% of those are likely immune. Many others are neurodegenerative. And the majority of patients suffer from around 300 diseases, but there are 5 to 6 that actually suffer from 7,000. The resources to work up a more rare disease are similar to those to work up a less rare disease, and approval is not streamlined to accommodate for the low numbers of patients who suffer, yet the need for treatment is the same. We need to do better. This is a list of all the genes that are known to cause autoinflammatory diseases, compiled in the INFEVERS database. In gray, have not been added. And you can see that the majority of genes actually lead to interferon-driven conditions, and the ability to do whole exome and whole genome sequencing has actually propelled the discovery prior to 2011, when this became affordable. Of those diseases, about a little bit more than a quarter are de novo mutations that actually lead to those diseases. De novo mutations are rare major causes of severe early genetic disorders, not only in inflammatory diseases, but also in many developmental diseases. And unraveling key pathways helps, or unraveling their pathways helps identify key regulatory molecules. And sequencing has provided next-gen sequencing tools to discover de novo mutations, identify their origin, and stratify risk. So in essence, TRIO analysis involves genetic analysis of father, mother, offspring. There are many copies of the genes that can be quantified. You actually have 50% from father, 50% from a mother. If the variant comes from a parent, it's present in the off gene. If it's not present in the parents, it is de novo. We've been able to get better discovery of de novo mutations by simply increasing the reading depth so that variants don't get actually sorted out as being errors. So de novo mutations arise as a result of errors in genome replication and repair of DNA damaging. And that can occur during parental embryogenesis, but also during parental gametogenesis, and also in the postzygotic offspring. The majority of mutations occur during parental gametogenesis. And that actually points to the risk factor, which is advanced age. And particularly advanced paternal age. In a 20-year-old male, the sperm cells have undergone approximately 100 to 150 mitosis, and spermatogonal stem cells undergo mitosis throughout life. That means they acquire about 23 mitoses every year. In females, the oocytes of 2 million when women are born decrease to about 200,000 at the time of maturation, but do not undergo any further mitosis. So new mutations that occur in women due to maternal age are much less compared to those that actually occur in men. So there are 3 conditions that are de novo that I have been involved in and that have given us targets for drug development and provided large insights. In general, it is important to recognize symptoms of patients with these diseases. They're often sporadic. If they are de novo, assess the extent of the organ inflammation damage and rule out concomitant immunodeficiencies, make a genetic diagnosis early, and I think this needs to be stressed to third parties or providers, characterize the immune inflammatory dysregulation by using biomarkers and initiate early treatment. And as Dr. Gedik had pointed out, she actually was coordinating an effort, or she was leading the interferonopathy effort to standardize workup and treatment for interferonopathies. And that is also out for IL-1 and will help physicians make diagnoses and treat patients. So my first patients and how I actually got into autoinflammatory diseases presented with an urticarial vasculitis with hearing loss. He had hearing aids at the age of 6, and he had bony deformities. And when we biopsied the urticaria, it was neutrophilic. That's rare because most forms of urticaria are allergic and they are caused by adaptive immune cells and not by innate immune cells. At the time when I saw this patient, Hal Hoffman had actually, through genetic analysis, discovered the cause for familial cold autoinflammatory syndrome. These are large families who in an autosomal dominant fashion have inherited mutations in NLRP3 in the NACH domain. At that time, whole exome and genome sequencing wasn't available, so we just had to do a candidate approach and hypothesis maybe these patients could have de novo mutations. We sequenced the gene and in fact found that this patient had a de novo mutation in the NACH domain, which basically pointed to the fact that he was on the severe end of a spectrum that was familial and intermittent due to cold exposure, but led to actually a permanent inflammation at its more severe state. It also provided insights into pathogenesis because basic research had linked inflammasome activation of that molecule. It wasn't called inflammasome at that time, to IL-1 production. And soon thereafter, the term inflammasome was coined when the components that led to IL-1 activations were found. The organs involved also included CNS with a neutrophilic leptomeningitis that causes headaches, and mild synovitis and bony enlargement, particularly of the kneecap, but also of the growth plate areas. And a multidisciplinary team is usually needed to treat those patients. This is actually, these are pictures from a beautiful tiny video produced by Kate Schroeder at Australia, but I wasn't quite sure if this worked, so I decided to actually make photos of it. It shows NLRP3 molecule here, which upon danger signal straightens out and then actually aggregates to form a nice flower-like structure that then polymerizes as filaments that actually form large branching structures on which caspase-1 molecules, an enzyme, attach that when activated and attached cleave cleave pro-IL-1 and IL-18 and another substrate, Gazdermin D. When Gazdermin D, and actually these little filaments form clusters and become factories of activating these three substrates. And Gazdermin 3, when activated, forms little rings and pokes hole into the wall of a cell, which allows IL-1 and IL-18 to be released. Usually these holes can get repaired, but in overwhelming infections, when the holes are large, the cell actually dies by pyroptosis, such as in infections, and then gets actually eaten up by macrophages. So the use of IL-1 blocking agents was hypothesized to help patients, and it didn't really take many patients to make that observation. Who's ever treated patients with CAPS will actually attest to the immediate response of resolution of rash, resolution of uveitis, inflammation of the eye, as well as CNS inflammation. And the responses, actually conjunctivitis, I'm sorry, responses to IL-1 blocking drugs are long-acting, and the safety profile of these drugs has been good until recently, after many years, when we've actually observed hard lumps that didn't actually disappear anymore, to show that anakinra amyloid can actually deposit at the sites of injection, particularly in patients like this one, who is not rotating the injection sites. There seems to be a late effect. We have early evidence that this can actually disseminate. So this needs to be watched and really raises the question that we need to get better treatment. The three structures, including anakinra, which I showed, which is a recombinant protein that binds the IL-1 receptor and prevents signal transduction, and two other drugs, rilonacept, which is a recombinant IL-1 receptor that traps interleukin-1 alpha and beta, and canakinumab, an antibody that specifically targets interferon beta, have been approved by the FDA in the United States and the EMA in Europe for the treatment of the entire spectrum of CAPS, and canakinumab also for periodic fever syndromes that are IL-1 mediated, and more recently for DIRA. The cost of this treatment is high, with a yearly cost of between $500,000 to $240,000 and a cumulative cost over 60 years that can go up to $14 million, which again points to the need of finding better and cheaper treatments for these patients. There are currently 4 inflammasomes that activate caspase and IL-1 that cause human diseases. And in all of those conditions, IL-1 drugs are approved. In NLRP3, which is the molecule here, the pyrin domain and the leucine-rich repeat domain, which forms this hook of the flower, are extensively modified post-translationally, and the mutations affect post-translational sites that often protect spontaneous aggregation, which, if mutated, leads to the aggregation in patients, and drugs are targeted to prevent the aggregation of that flower-like inflammasome that triggers and activates caspase. And 2 drugs are currently in clinical trials. One is tranilast, a tryptophan analog that inhibits the formation of the inflammasome aggregation, and another one that Novartis is sponsoring, DFV890, and often the function of this molecule is not revealed. So, but all of those target the oligomerization of NLRP3. Another strategy that has worked in mice is to block gasdermin itself. There are currently no drugs, although disulfiram, a drug that is used to treat alcoholism, actually has shown to block gasdermin D. Again, I would not recommend to treat patients with this, but in mechanistic studies, this has worked. So in summary, NOMAD, sorry for misspelling, is a severe CAPS, is on the severe spectrum of CAPS. It is an autoinflammatory disease caused by gain-of-function mutation in NLRP3 that alter the threshold of caspase activation and IL-1 cleavage. It's a critical regulator of responses to endogenous and exogenous danger signals, and has become the target for drug development, as activation in the tissue of the NLRP3 inflammasome has also been shown to drive coronary artery disease, atherosclerosis, and neurodegenerative diseases such as Alzheimer's in mouse models. NOMAD also is a model, or has become a model, to validate NLRP3 activation in human disease and serve as a proof of concept for new drugs. The second group of conditions actually started with a referral of a girl who had peripheral vascular disease and was unresponsive to anakinra and presented with fevers. Her disease is very rare. She actually had a persistent type 1 interferon signature. The history of conditions with high interferon signatures goes back to Aicardi-Gouttiere syndrome, where for many years, a torch-like encephalitis was diagnosed by looking for interferon alpha activity in the spinal fluid of patients with this condition. That patients presented with basal ganglia calcifications, often in utero, and progressive spasticity, mental retardation, and often also microcephaly. And it was Yannick Crowe who in 2006 started unraveling the pathways that actually lead to the activation of the interferon signature by showing that recessive loss-of-function mutations in TREX1, an endonuclease that actually processes self and viral DNA, is actually accumulating or is the cause for those diseases, and then fueling research that showed that the inability to process intracellular nucleases leads to their accumulation. And at that time, the viral sensors were being discovered, were triggering the interferon signature through signaling through viral sensors, which basically also told us that it is not just viral DNA and RNA that triggers the viral sensors, but also self-DNA and RNA. This has been a major impact, and Yannick coined the term of autointerferonopathies as a driver of disease, assuming that it is type 1 interferon that fuels the disease. At the same time, we actually had struggled with another disease, children who presented with panicolitis and loss of fat lipoatrophy, systemic inflammation, who also had basal ganglia calcifications, which raised the question of interferon. Those patients, the first ones, had mutation in PSMB8, which was actually discovered by 3 groups simultaneously, and we had identified a chronic interferon signature. And in 2014, we've used for the first time TRIO analysis, and SAVI was the first disease that was actually identified by finding de novo mutations in an autoinflammatory disease. So as mentioned, CANDLE and SAVI express an interferon response gene signature that was described by Virginia Pasquale's group in lupus patients in 2003. This was in contrast to NOMAD patients and to healthy control patients. And genes out of the signature are used to probe for and quantify the interferon response gene signature. These are 6 genes. You can't see the name. I'll show them to you later, that Yannick Crowe uses. There are different subsets used for scleroderma or for lupus. And we've actually used a very reproducible and fast way of quantifying genes, nanostring, and have chosen to use 28 genes, which includes the 6 that are commonly used or the 4 that actually have currently been used in the TULIP studies in lupus. So they also are quite homologous and do actually are upregulated and can be used to monitor treatment. So when should you think of SAVI? SAVI is caused or presents with a vasculitis in cold exposed areas, which is in contrast to lupus, which basically leads to a rash, a malar rash upon sun exposure, and also shows immune complex deposition at the dermal epidermal-dermal junction. And many patients actually have features of chilblain-like lesions or vasculitic lesions in areas exposed to the cold on the floor when the foot steps on the floor, or apical surface areas. There's progressive autoamputation due to gangrene, which can occur more chronically as a result of Raynaud's, that is chronic, or acute here in a baby as a presenting feature. SAVI can be sporadic or familial, and I think we are probably close to over 100 patients, because patients with lung disease who don't have peripheral disease usually do not get diagnosed until much later in their life. And this is something to screen for. We actually had 2 referrals of young men with end-stage, very advanced lung disease within the last 3 months. So other features include systemic inflammation, and patients usually have autoantibodies, but they don't correlate with the disease activity, and we typically don't see immune complexes deposited in the affected organs. Cytopenias are also common. The mortality untreated is high, and the cause mainly is lung disease. Some of the cytopenias respond when patients are not flaring or increase during less active disease, and growth delay is a common feature that we see. Most of the mutations are in exon 5 of gene that previously was called TMEM173, and now actually gets its name STING, which stands for stimulator of interferon genes. The mutations that cause SAVI are depicted here. The 2 mutations most common is V155M, which presents with more severe lung disease, and over almost a third of the patients have minimal peripheral vascular disease, which makes a diagnosis often late, while patients with the N154S mutation have more peripheral vascular disease, and their lung disease can be delayed or somewhat milder. James Chen's group in 2013 had solved the structure of STING by using cryo-electron microscopy, and had found that it dimerizes. And the mutation, so that these are the 2 dimers that form, and the mutations are actually in the neck domain. This is the binding domain. And physiologically, STING gets activated through dinucleotides that get released by cGAMP, sensor for viral double-stranded DNA, and that when activated releases dinucleotides. There are some bacteria that release dinucleotides and can directly activate STIM. So upon binding, the binding head rotates 180 degrees. So green moves to the left, and the molecule itself closes and forms a bridge with the C-terminal end, which basically then allows the recruitment of a kinase TBK1, which phosphorylates these tiny ends, and I'll tell you what happens. The mutations in the binding area here in the connector helix loop area weaken the neck and actually allow for spontaneous rotation in the absence of binding and oligomerization, which basically activates downstream pathways. But there are other mutations like here, which hold the end of the C-terminal tail that when mutated, release that. And our predictions actually been Lynn in my lab, who has characterized the mutations, believes that there are changes in the steric hindrance that prevent oligomerization with wild-type sequence that are prevented by the mutation or abolished by the mutation, so that the mutation can spontaneously form oligomers without rotation and closing the binding site. So when TBK1 is recruited, phosphorylates the CTT tail of STING, it can recruit IRF3, which is a signaling molecule that forms dimers, transmigrates into the nucleus where it drives the transcription of interferon beta. And so when interferon beta gets released, it distributes and activates surrounding cells through the T-cell receptor. It can actually activate itself also through the T-cell receptor. The signal is mediated by the JAK-STAT pathway and then leads to the transcription of the interferon response genes that we measure as a readout for type 1 interferon signaling. So until recently, the only strategy to block interferon signaling have been the JAK inhibitors that inhibit the JAK-STAT signaling pathway of type 1 interferons. Unfortunately, they are actually approved for the treatment of rheumatoid arthritis, and they work in rheumatoid arthritis because there are multiple inflammatory receptors that share the JAK-STAT pathway as a signaling pathway, including IL-6. But also, it is approved for polycythemia vera in myelofibrosis, where it actually likely works by blocking erythropoietin signaling, which also uses a JAK-STAT, which basically points to the fact to the fact that we see unwanted effects in these patients. However, when we treat candle patients, 50% of those patients go into remission. Their growth parameters improve compared to pretreatment, and the cytopenias resolve. However, the drug exposure is much higher than treating rheumatoid arthritis patients. It's actually 3.6-fold higher than the dose approved for the treatment of rheumatitis in the United States, which is only 2 milligrams per day. And the side effects that Dr. Gedig actually described pretty well are the reactivation of BK viruses and other viruses that really limit their use and warrant monitoring. However, we do have proof of concept that the interferon signature normalizes in those children who remit. However, SAVI is different. Although we see some improvement in daily diary scores, lung function, and also in the control of the peripheral vascular disease, the interferon scores do not normalize in these patients, which basically points to the fact that we still can't answer the question that is posed on whether we are not blocking interferon well enough and that interferon signaling is driving the disease, or whether STING, which in mice seems to be more activating NF-kappa B and less IRF3, has a function that we are not blocking with the drugs and that we need to actually block other targets. There are differences in the interferon signaling pathways in mice and men, and there is actually quite a bit of sequencing difference in the molecules that determine this pathway, which does suggest that the contribution of interferon to the disease in the mice and men might be different. Also, the murine model of STING does not reflect the lung disease in patients. This is a really sad example of the progression of lung disease in a SAVI patient on the JAK inhibitor. The patient presented in 2020 on 2 liters of oxygen at the age of 2. She was started on JAK inhibitors, which actually improved her lung disease. She was off oxygen, but then developed SARS-CoV-2 COVID at the age of 6. This is her lung at that point. She cleared her lung, but then developed aspergillosis, which initially was fumigatus. She was treated, and then Kalydeustos and a cryptic aspergillus did not respond to the treatment, grew. She developed huge pneumocele and actually passed away. This is a patient with a V155M mutation, which is an example that we need to do better. In addition to anifrolumab, which actually is approved for lupus and can block interferon signaling specifically, which has really not been extensively enough tried or has not been used in SAVI patients, but is being evaluated. There is another opportunity, and that is small molecules that actually inhibit STING aggregation. And we've tested some in vitro. They do work in vitro models, but their safety has not been proven yet in human models. So we'll keep ourselves. We'll be waiting for this to progress. So SAVI is an autoinflammatory disease caused by gain-of-function mutations in STING that alters the activation threshold and activates downstream pathways, including interferon beta signaling. STING is a critical regulator of responses to microbial and cell double-stranded DNA and the STING pathway, and has become a target for the development of therapeutics. SAVI is a model disease to understand the role of the effect of STING in human pathology. And there is actually data suggesting that it not only activates monocytes, which actually produce the majority of interferon beta in SAVI patients, but that it also has an impact on the fate of endothelial cells and cells that we don't necessarily target by the treatments we currently use. The use of novel treatments will tell us about the role of type 1 interferons, and the disease may help us identify STING-specific targets. So untreated autoinflammatory disease can often lead to organ damage that is really progressive. And so far, repurposing drugs has focused on quenching the inflammatory response of innate immune cells. But as just mentioned, some of the mutations, and actually less the inflammasome, but the mutations I'm talking, particularly STING and the one I'll be talking about, have cause cell intrinsic dysregulation by affecting the cell death or differentiation of the cells, such as endothelial or epithelial mesenchymal transformation or senescence. So there are few treatments that actually focus on rescuing cell fate or changing cell fate. So the disease that I'm going to talk to you about now teaches us about dysregulation of neutrophils, but also of endothelial cells. And like in many of the models or diseases I described, the animal models do not accurately reflect the organ damage, which led us to use iPS cells and develop actually cell models or cell line models to investigate the pathogenesis. So that story started with a third patient who actually was sent to us when he was barely 2 years old. And his disease is ultra rare, with likely less than 10 or 20 patients with exactly that disease worldwide. He presented around birth with fever, splenomegaly, purpuric rash, and severe thrombocytopenia. He was splenectomized at the age of 8 months. And then was basically developed more prominent periorbital erythema, testicular swelling, abdominal pain, anemia, leukocytosis, and thrombocytosis, all features of small vessel vasculitis, and a biopsy showed a neutrophilic vasculitis. When he presented at the NIH, he actually had low titer autoantibodies to ANA, anti-Smith, anti-SSA, anti-cardiolipin, anti-mitochondrial, and a rheumatoid factor. And a liver biopsy actually showed vanishing bile duct disease and bridging fibrosis. I'll show you this on a later slide later. So the differential diagnosis when he came had already been ruled out. He didn't really have immune complex deposition anywhere in any of the immune-mediated conditions of childhood had been ruled out. He did not have leukemia, and so he came to us for workup of an undifferentiated autoinflammatory disease. And again, using trio analysis, we found that he had a de novo mutation at a kinase, Lin kinase, a known Src kinase, at a critical tyrosine position 508. And it rarely happens that a mouse exists at the time when we actually find a mutation, but a gain-of-function Lin mutation had been described at exactly the same position in the mouse where our patient had a stop codon. So the critical tyrosine was here modified by phenylalanine, and it was abrogated in our patient. Now, the mouse developed glomerulonephritis, anasarca, high titer autoantibodies, and is studied as a model for lupus. But the mouse has no skin disease and no liver disease. So we still explored initially the possibility that that is an autoimmune disease and treated him with IVIG and pulse methylprednisolone. The antibodies disappeared and actually never recurred in him, but the transaminitis persisted and the liver fibrosis persisted. So the mutation gave us a clue because Src kinase inhibitors are used in oncology and are approved. And Dasatinib has proved inhibitory function in Lin kinase. So we initially studied his effect of Lin kinase in B cells, and Lin kinase itself was activated here in blue more than in his parent, and an inhibitor of Lin kinase downregulated phosphorylation, but also targets of Lin active kinase that get phosphorylated by active Lin kinase, such as CD19 and others, were increased and were normalized by use of Dasatinib and another more potent Src kinase inhibitor that is just a chemical not approved for patients. We actually enrolled him in a compassionate use study and in the NCI, and he was treated with Dasatinib, and we observed that his inflammatory markers, which were high and only responded intermittently to steroid pulses, normalized. And when we withdrew Dasatinib, they actually increased again. And when we restarted it, he normalized the values. He did have continuous flares when he had infections, but he was much better manageable. And we had shared the mutation at a conference. And in 2017, we referred a patient actually from England, from Paul Brogan, who had added that gene to the autoinflammatory disease panel and identified patient who at that time was 15 and had presented with small vessel vasculitis. He had been empirically treated with a TNF inhibitor and had resolved his rash, but he did not have liver disease. That led us in limbo whether or not the patient's, whether this mutation is causing liver disease. Interestingly, this patient had the exact mutation that had been made in the mouse, and he also did not have evidence of autoantibodies, which had been screened, which he had been screened for regularly. And then at the end of 21, we actually were referred a third patient, and that patient actually also had a missense mutation that led to a nonsense mutation that basically led to an abrogation for stop codon, which basically eliminated the last 6 amino acids. And here we basically, in our index patients, we lost the last 5 amino acids. In each case, the critical tyrosine was actually altered. This patient did present with hepatic fibrosis, which basically suggested that the residual molecules that are actually quite conserved among different Src kinases likely allow still some negative downregulatory effect and may therefore not lead to the liver disease we see with a more severe truncating mutation. All patients, all 3 here in the 3 colors, red, yellow, and blue, had repeated elevation of their C-reactive protein here compared to NOMID and SAVI, and also markers of neutrophil activation like soluble L-selectin as well as soluble ICAM, which also is a very important marker of endothelial cell activation. And that led to in vitro studies. First, wild type LIN is inactive. The tyrosine that is mutated in these patients is phosphorylated and basically closes the kinase domain. Upon dephosphorylation, another tyrosine in the kinase domain gets phosphorylated, and that constitutes the active form. If the negative regulatory domain is gone or altered, then a constitutive phosphorylation of tyrosine 397 leads to constitutive active kinase. And we tested that by transfecting the various constructs into HEC293 cells. And as expected, there is no phosphorylation of the inhibitory tyrosine, but there is increased phosphorylation of the activating tyrosine. And we were quite concerned because of the progressive liver fibrosis. Here is a Mason trichrome stain, which in blue shows areas of bridging fibrosis that actually had progressed on IVIG and glucocorticosteroids, and actually led to the initiation of Dasatinib, which basically showed reduction of the fibrosis with treatment. We've also seen bile duct recovery. He had lost 50% of bile ducts in the portal triad on treatment and basically recovered those on Dasatinib. And furthermore, his liver enzymes and gamma GT normalized on the treatment. We started etanercept treatment when we heard about the second patient and had started him on monotherapy, but it actually led to more flares of his skin and also to some rebound of his inflammatory markers, which basically led us combine Dasatinib and etanercept, which is the treatment he continues to be on. And we've also seen elevation of adhesion markers in neutrophils with high beta-endocrine expression before treatment that dropped on Dasatinib and then was best controlled on the combination. We've seen a flare in the neutrophil markers when he's been treated with etanercept alone. So how does this fit in with the generation of fibrosis? LIN this is actually a portal or is part of a liver lobule and shows the portal triad. Here's the portal triad of the portal vein, the bile duct, and the hepatic duct. And LIN is expressed the cholangiocytes, which basically had disappeared on before we treated him and caused the vanished bile duct disease, but also in Kupffer cells and in hepatocytes, and there is a layer of endothelial cells that is fenestrated, and the liver sinusoidal endothelial cells are LSECs, basically are in the sinusoids and screen blood that is delivered from the portal vein. In instances of liver injury, they actually get activated and release substances that activate stellate cells to differentiate into myofibroblasts, that lay down extracellular matrix and lead to the production of extracellular matrix in the sinusoidal canals, which is what is seen in bridging fibrosis. We had 4 liver biopsy samples and conducted RNA-seq and have seen that on treatment, markers of epithelial surface actually increased, corroborating the recovery of the bile ducts on treatment. And at the same time, adhesion molecules, extracellular matrix genes that express these molecules or these proteins, and inflammatory cytokines and chemokines decreased, as well as LIN kinase message itself, and markers for immune cells. And we tested a scar-associated liver signature that had been established in mice and had been tested in alcoholic liver cirrhosis in adult patients. And that signature actually decreased, suggesting less activity of LSECs and also mesenchymal macrophages that are driving the fibrosis in the liver. But the hallmark of this disease has been the neutrophilic vasculitis. We've seen neutrophils surrounding the destroyed vessels that are positive for LIN, and LIN staining is seen in endothelial cells as well as in the monocytes surrounding it. There are less macrophages in the skin. The affected vessels in the patient compared to the control stain positive here in red for ICAM, meaning they are activated, and staining for nets shows that the neutrophils form nets around the tissues that are destructive and likely destroy the vessel wall. Here, destruction of the vessel wall in a patient compared to the line that you actually see for here using von Willebrand factor of an intact endothelial cell. So these findings suggested that the migration of inflammatory cells through the endothelium might actually be mainly impaired and regulated by LIN kinase, and raised the question of the role of endothelial cells. To do that, we collaborated with Manfred Böhm's lab, and Quibin and Dan made iPS cell-derived endothelial cells, IECs, and also so mutant patient IECs, but also CRISPR-Cas gene-corrected patient cells that are isogenic, he called isogenic IECs. And they were compared to healthy control IECs, and we used healthy control neutrophils in co-cultures to separate the effect of endothelial cells on this pathway. And what we observed in co-culture experiments is that neutrophils adhered to the mutant endothelial cells much more than in the isogenic genetically corrected cells, which pretty much behave very much like controls. The adhesion was blocked by Dasatinib, but not so much by the TNF inhibitor, and there was no synergy when both drugs were combined. We then overlaid neutrophils over a monolayer of mutant and isogenic endothelial cells in a Boyden chamber and measured the transmigration of these cells through that layer, and have seen that it is the neutrophil migration through that layer is much increased through mutant cells, and it is completely corrected when the genetic mutation in the patient is corrected. In terms of treatment, Dasatinib and TNF inhibition in both instances decrease the migration, and again, there is no synergy. And we had observed that in areas where neutrophils overlay an endothelial layer of cells, here the connection of the endothelial cells is shown by VE cadherin, an adhesion marker, had loss of these markers on the surface. And that suggested that there may be a cell-cell barrier defect. And that can be assessed by looking at transendothelial electrical resistance, which allows to assess vascular permeability. And when we overlaid neutrophils here in red over mutant endothelial cells, we've seen a decrease in the electrical resistance, suggesting leakiness, which was not seen in the isogenic control. And as had actually been seen before, Dasatinib does not correct that leakiness or the transendothelial electrical resistance, but the TNF inhibitor actually does that. And that actually suggests that Dasatinib likely has off-target side effects, because targeting LIN directly by the genetic corrections did actually fully correct the defect. And it points to the fact that there might be benefit in generating targeted treatments. So in summary, de novo mutations in LIN kinase cause a novel autoinflammatory disease, LIN-associated vasculitis with liver fibrosis, that we called lovely. LIN kinase is a key modulator of beta endocrine function on neutrophils that mediate adhesion to endothelial cells. and also of regulating ICAM exposure or activation on endothelial cells. Lin gain of function mutations result in increased neutrophil adhesion, transendothelial migration, and microvascular permeability that lead to small vessel vasculitis and hepatic fibrosis. And combined treatment with Dasatinib and etanercept improves small vessel vasculitis and reversed liver fibrosis. And neutrophil endothelial co-culture models allow for the assessment of drugs that can modulate different aspects of endothelial function in neutrophil recruitment and diapedesis, and identifies Lin kinase as potential therapeutic target in non-syndromic small vessel vasculitides and inflammation-induced liver fibrosis. We've actually started to work with somebody actually at Pittsburgh who is trying to make a specific blocker for Lin kinase. So in summary, I've introduced patients with 3 de novo mutations, which all have provided clues to important targets in regulating IL-1 activation, interferon beta, and other downstream STING activation pathways, and by amplifying the signaling that Lin kinases actually amplify on cell molecules. And in each case, they've been contributing or have become targets of drug development, including specific NLRP3 inhibitors, gastromin D inhibitors, STING inhibitors, and selective Lin kinase inhibition. I may have time, I thought I actually removed it, but there is another disease that, first there's a 4th patient with a Lin kinase mutation, actually a girl, which is expected, who basically presented with very similar findings. She also has other features suggestive that there might be additional genetic factors that play a role in her disease, including a big forehead, which is not seen in the 3 other patients. And there is a homologous kinase, HCK, which is regulated very similarly with a negative regulatory tyrosine kinase. And this patient, again, had a deletion of that regulatory component and does present with a small vessel vasculitis. But interestingly, that molecule is higher expressed in macrophages than in neutrophils and leads to a very severe lung disease that actually led to death in that patient. So there are other Src kinases that do that. So to conclude, the genetic approach to the exploration of rare autoinflammatory disease phenotypes continues to provide insight into pathomechanisms that cause systemic and organ-specific immune dysregulation, which continue to reveal novel treatment targets, and in some instances, deliver precision medicine to those patients with rare diseases, as shown in the last part. This is the current team, and I'd like to thank Adriana de Jesus for the genetic workup and patient care bin, and Sarah for seeing patients, and also Dr. Chedingedic for her work when she was in my group on patients, on identifying or basically assessing the safety of patients of JAK inhibitors and the treatment of patients with INFEVERS, and really driving the consensus work on treatment recommendations. There are many to thank, and on the last example, particularly our collaborators in the Czech Republic, who have reproduced or have actually done many of the activation work in their lab because the patient was too small to travel, and the referring team in Great Britain, and most of all the South Carolina group, who has been taking tremendous care of the index patient, and the many others who have contributed to the cohorts of patients that I have presented, and to the NIH collaborators that form the multidisciplinary team that helps clinically evaluate our patients, and to my funding resources. So thank you very much for your attention, and I'm happy to take any questions.